lipids, they can modify the protein, and they can alter the chemistry and biophysics of the proteins in that environment, whether it's the proteins themselves that are glycosylated or the proteins that exist in that environment. And glycobiology and glycans are largely understudied in the context of biophysics. So if we were just go on PubMed and query every one of the macromolecules, if we get proteins, we get 8.9 million hits. Nucleic acids, we get 3 million hits. Lipids, 2 million hits. Glycans are understudied with only 700,000 hits. So why is this? Well, that's because glycans are heterogeneous and they're non-templated. They're chemically difficult to synthesize. They're highly dynamic. And unlike molecular biology, where you can clone a gene or manipulate DNA, express protein, we do not have those tools available to us for glycobiology. In fact, if you think about common things like a Western blot or a Northern blot in biochemistry, where you probe a specific DNA presence or a specific protein presence, those tools are completely absent in glycobiology. They just don't exist. And the main textbook for this, the first edition for it, Essential Glycobiology, was just in 1999. And that's kind of evolved the latest edition in 2022. So we can't talk about glycobiology without talking about two essential people. One being Ajit Bharti, and one being Karan Bartosi. Does anybody know anything about Ajit? He's actually here. So Ajit's winding down his lab. He's in the process of retire, and unfortunately he has Parkinson's. But he was the founder of the Glycobiology Research and Training Center here at UCSD, and has made a very significant impact in glycobiology, both here and internationally. That he is probably the most respected name, to the point where when I put it in ChatGPT, he came up first, and the Nobel laureate Karan Bartosi came up second. So that's pretty cool. He's here. We have a strong history of glycobiology here, and a lot of it's because of Ajit. Okay, so these glycopolymers, they do a whole bunch of things. So they provide information between cells, and from the extracellular space to the interior, you can label proteins for transport to specific organelles based off the absence or presence of a glycan, or for destruction. So if your protein's misfolded in the ER, it can be glycosylated in a certain way, and that will send it to the lysosome for degradation. They can serve as recognition sites for extracellular molecules, or parasites, or viruses, but they can also act as a barrier for microbes to prevent them from getting infected. And in most cases, what we'll talk about are these carbohydrates as they're pinned and covalently bonded to proteins or lipids. So far, we've talked a lot about sugars looking like this. And this is a Mod 9 glycan. So you can see that there are 9 mannose residues, and they're attached to these core 2 Glycan structures. This would be way too long to draw on an exam in chemical form, right? Like, that's a ton of atoms. I think that would take me even 10, 15 minutes just to draw that. So is there a simpler way that we can represent these sugars but still retain a lot of this information? So Ajit and colleagues actually came up with what they call symbol nomenclature for glycans, where we take geometric shapes and colors and pair them to different sugar types. So you can see here, for example, these glycanacs are these squares, these mannoses are these green circles, lactose, a yellow circle, a galnac, a yellow square, and so on and so forth. So then, essentially what this does is it takes these complicated structures and allows us to more or less reduce them into geometric shapes where we can have a key to read out the different aspects of them. In this, it does have linkages here in the dashed lines. Those are not always included. Actually, they're very much not included. I don't think I've ever really seen that. But in general, you can get an idea of the bonding structure. Shown here is the Mod 9 glycan in chemical form, and shown here is the Mod 9 glycan in its symbol nomenclature for glycobiology form. Yeah?
Are we expected to understand the individual structures that are in that key there? Like galactose and everything? Are we expected to memorize stuff like that?
I would not ask you to memorize it, because I do not have this chart memorized, right? So if I were to give you this chart though, maybe as an appendix page, then I could ask questions with it.
But like, would there be like a labeled chart of like what those molecules look like as well, or would we have to know that?
You're saying going from like a mono to
Yeah.
Will you post the updated slides?
yeah. I update the slides literally at midnight the night before. So I'm working on these pretty much until this. So I'll post them at the end of the class. Okay. So these types of glycans come in two major forms. You can have N-linked glycans and O-linked glycans. And N-linked glycans get added to the side chains of asparagines, and O-linked glycans get added to the side chains of serines and threonines. So these are two major classes of glycans, but then they get further diversified and you have further substructures in each class. Shown here are the three major classes of N-linked glycans going from high monos to hybrid class. Shown here are just a few different structures of O-linked glycans where you have a linear chain or you can have these branch chain structures. So, if we think about N-linked glycosylation, N-linked glycosylation occurs at this specific sequence. Acin X and then either a threonine or a serine, where that X is any amino acid besides a proline. What happens is that the anomeric carbon bonds with the acid side chain amide, and this is done by an N-acetylglucosamine, and it is in the beta anomer. So you have your glucosamine, you see here the beta anomer of its hydroxyl group, and you see an asparagine side chain, they come together to form this linkage. Okay. It is not that simple, because while this is the zoom in, they get added actually as these mod-9 glycans and further processed and tailored enzymes. So the way this process works is you start off in the ER and your polypeptide is being co-translationally translocated. You guys, are you familiar with that from cell biology? If you're not, the ribosome makes membrane proteins, or proteins that are secreted, in the ER by bringing the mRNA to a translocon, and then essentially the exit tunnel feeds the polypeptide into the ER. As this happens, oligosaccharyltransferase transfers the high monos glycan onto it. This glycan then acts as a handle for the calnexin and calreticulin chaperones that help the protein fold. And it acts kind of as a lever arm to like a pull on this polypeptide until you get the proper folded structure. At this point, the glycan also acts as a signal to whether it's going to go be degraded or whether it's properly folded and can move on. This then gets sent to the Golgi. In the Golgi, enzymes see that glycan and do further tailoring to it. And they trim it back in the cis Golgi to a mod 5 glycan. As this then passes further into the medial Golgi, it can be either elaborated on by things like mucosal transferases, it can be cut back further into a mod 3 glycan and then modified, and then it can be elaborated into different structures. And this is what results in either hybrid or complex glycotypes, where you have the trimming of a high monos glycan down to its either mod 5 core or a mod 3 core, and elaborated on into hybrid or complex glycotypes. So this is all done enzymatically, and this is what dictates whether it's going to be a high monos, how it was added, or whether it's going to be processed into a hybrid or a complex type. Generally, we think of this as these being the least processed and these being the most processed, with hybrid being in the middle along the way. Okay, unfortunately, glycobiology gets much more complicated at this step. And the reason why is because within each of these classes, there are tons of substructures. So if we just look at high monos glycans, we have mod 9, which is shown here. But then we also have mod 8s, and there's 3 different mod 8s depending on what sugar gets removed. Then we have mod 7s, where there's 3 mod 7s. And there's mod 6, mod 5, and mod 3. And these structures for mod 6, mod 5, and mod 3 are the most biologically relevant ones. There's also, MON3s that just go up this branch. Or there's MON5s that have another molecule out here. So you can see that you get these structurally diverse classes based on how the enzymes act on these molecules. Further complicating this, organism to organism, there's a ton of variation in glycans. So if we look at N-glycans from yeast, they have these massive oligomono structures that we call monods that can go on for hundreds of monoses attached to them. But if you look at insects, we just have this small posse-mono structure where you have this MON3 core structure. Other mammals form these very bifurcated complex glycans, and then plants and humans have their specific glycotypes as well. If we were to look at all the structures from biological tissues of glycans that have been purified, we get something like this. And I don't even think this is comprehensive anymore. So you see that this is a massive heterogeneous pool, and this is just of the N-linked glycan subtype. We still haven't even talked about O-linked glycans. So, needless to say here, glycans are heterogeneous. That leads to two terms that we need to define here. We have macroheterogeneity. So if you have a peptide that has an N-linked glycan site, we have this acid RH there. It can be glycosylated or non-glycosylated. So the presence or absence of a glycan at that site is what we term macroheterogeneity. Okay? We also have microheterogeneity. Microheterogeneity says that site is occupied, but it might be occupied with a bunch of different substructures. So, in one protein copy, it might have MON8, and in another protein copy, it might have MON7, based on how this was processed by the enzymes. Does that make sense?
Sorry, so the original one is only high monos. The
All-
macroheterogeneity.
All N-linked glycans get added as high monos.
Okay.
So macroheterogeneity, you can think of it for N-linked or O-linked, just describes is a glycan there or is it absent. Microheterogeneity is a variation of the structures. So what this might look like in a real world example is something like this. This is RNase B. RNase B has a single site of glycosylation and it's got high monos glycans. If we do glycan release or mass spec profiling, what we see first by the LCMS is the presence of 5 glycotypes, and we see 5 different N to Z ratios. These correspond to the different types of glycans present. So you can see in a population, you have this weighted distribution between a lot of MON5, some MON6, a little bit of MON7, some more MON8, and a little bit of MON9. So this is what we describe as microheterogeneity. And this is described then on a single site. So, in a population, we have this mixture between MON5 and MON6. The terminology for the difference between those two types of proteins are called glycoforms. So if you have a MON5 containing RNase B or a MON6 containing RNase B, they are considered different glycoforms of each other. Does that make sense? Cool. So we're going to talk about spike here for a second because the spike protein from coronaviruses is very well studied for its glycosylation pattern. So I think we've gotten to the point, like glycosylation isn't coded for. We can't just take a DNA plasmid, clone something in, and say make MON5 glycan, right? It is done enzymatically and it is done stochastically. This raises the question of will it be the same in every organism, and will the same protein always have the same glycan in the same location? Well, we can find this with RNase B that this isn't true. So spike again has N glycans, and you can see that these range from complex, being shown here in purple, or oligomonos in green. If we look at the per residue glycotyping, you can see the distributions of each glycotype at each of the 22 sites. So now imagine the number of glycoforms that are possible for this is every permutation of occupancy seen here. So this can demonstrate both microheterogeneity, shown here in N17, we have a couple high monostructures, this would be 7, 6, and 5, and then you have a hybrid structure. And then you can see down here, here, you have complex structures, or it's unoccupied. So this residue is seeing macroheterogeneity, and this residue is showing heavy microheterogeneity.
What determines how complex it's going to get, or whether or not it gets a glycogen?
So what factors might affect this? Great question. Any takers? If you're in the glyco class, you can answer this.
It's like presence of glycans and like how we had said like the safe one, how available it is.
So is the sequence for glycosylation present? Okay. Is it solvent accessible to the enzyme? That's what will dictate macroheterogeneity. When we look at microheterogeneity, we look at things like solvent accessibility, residency time in the ER Golgi, environmental conditions, pH, temperature, etc. And then the expression of the glycan tailoring enzymes in the organism that you are thus looking at. Does that make sense? So this is why you have this heterogeneous mixture of these glycans and why it is so difficult to study glycobiology because of this mixture. You can't just have one species and have a homogeneous sample and say, hey, this is how these interact. Okay, so now that we've talked about N-linked glycans, we can talk about O-linked glycans. O-linked glycans get added to the side chains of serines and threonines. So you have this hydroxyl group and you have the anomeric carbon. They form these structures where here is a threonine linked sugar, here is a serine linked one, and this is what we would refer to as O-linked glycosylation. Again, N-linked is called N-linked because it's linked to a nitrogen amide side chain. O-linked is linked to a hydroxyl group, so it is then called O because it's the oxygen. Okay. In this, O-linked glycosylation is actually much more familiar to you guys than you're probably aware of. You can form these various different structures. Again, they can be branched, they can be these one linear chains, or you can see they can form these unique structures that then have different things like phosphates in them. Here's ribitol phosphate. I think this is really interesting because in these structures, we get sugars incorporated that are not cyclic sugars. Ribitol, its structure this is a structure of ribitol. It doesn't have an aldehyde or a ketone in it. So it can't form the cyclic sugar structures. So it is actually in here as this linear structure. And you see it's actually in here as ribitol P. So it can have a phosphate and it's actually connected through a phosphate linkage to the next sugar. So O-linked glycans have a ton of weird but cool chemistry associated with them, where the N-linked glycans are a little more standard in that sense. O-linked glycans also are generally very, very long. They're either very short or very long in nature. They're not typically in these intermediate tens of sugar structures. Okay, so where can we see O-linked glycans in biology? We're all familiar with the ABO blood groups, right? These are glycan structures. So shown here is the A, and here's the B, where we now move. Oh, those look the same for A and B. No, sorry. You have a galactose instead of a galactosamine is the difference between A and B. And the presence of an additional sugar there in that branching structure is what is the difference between A, B, and O. So when you have a certain blood type, let's say B, you have this sugar there, and you can get O because you don't form an immunogenic reaction to that because that's already part of that core structure, right? On the same way, if you have A, you have that core structure of O, you just have a different sugar on it. So you don't have a reaction to it because that's how your blood's been evolved. If you have the mix of them, then you have both, and you can recognize both of them and have been evolved, your immune system has evolved to recognize both of them as yourself, not as something else. Does that make sense? So it's all sugar based. And that then leads us into this next conversation of sugars can act as places where, or as handles for proteins to attach. So what happens to blood if you mix type A and type O? Anybody know? We just have a bunch of erythrocytes, and we have this protein called leptin in here. Have you guys never done this? Like, you take a little bit of blood and you like do your own little blood types and like, no? I guess like since I was a kid, like health and safety things probably got more complicated. I feel like in 4th grade health we did this. Okay, so if you get blood from the wrong blood type, the leptin's in the blood, and a leptin is broadly defined as any protein that binds a carbohydrate. But there are specific leptins in the blood that will recognize the other blood type as not your blood. It will then interact with it, and as this happens, it actually interacts with all of the blood cells, and it agglutinates them. And thus you form these little clumps, and you actually kind of clot and coagulate your blood. This is why matching blood types is so essential to blood transfusions. If we look at this in an actual experimental setup, you might have something like this, where these plates that this is in, these wells look like this. So what you can do is you can fill this with erythrocytes, right? So you just have a bunch of red blood cells floating around here. Then you add what you think could be, let's say it's the other blood or something that might interact with it in a negative way, and it coagulates it. So now it forms this giant clump at the bottom of the plate. And that's what we're seeing here. That you're actually forming those clumps or agglutinating these things, or these blood cells.
How come that doesn't happen with like universal donors and universal receivers?
So universal donors and universal receivers. Okay. What is the universal donor blood type? O. So O, you have this 4 sugar structure here. If you look at type B and type A, that 4 sugar structure is present in both of them. So your immune system has selected out antibodies or leptins that bind specifically to this structure so they don't agglutinate your own blood, right? But they might not have selected against ones that recognize this structure or this structure, and actually we know that they haven't. So when you get something that you don't have a structure that recognizes, so the universal donor is O, right? Because it doesn't differentiate between type A and type B. The universal acceptor is AB, because you have both of the antigens, and the type O doesn't bother you. Does that make sense? So yeah, that's the molecular basis of blood typing. All sugar chemistry, which is pretty cool. There's also the rhesus factors that can get more complicated, the positive minus, but that's for another time. Okay. So the other thing is that glycans are highly dynamic. Shown here is a glycosylated viral protein. I believe this is actually the head of HIV GP120. You can see that these glycan structures are built into here. And for example, up here is a branched end-length glycan. This is a single snapshot of a molecular dynamics simulation. Molecular dynamics uses physics-based methods. So basically, F equals ma to push on these molecules and see how they move, right? So there can be a way to determine molecular motions. So if you push on these molecules and take a snapshot every microsecond or every nanosecond and see how they move, and then take all of those and combine them together, you can get an ensemble of every place that glycan occupied in the structure. Make sense? What that looks like is this. So because of the dynamics of the glycan, you now almost completely cover the protein surface. This is to show that these glycans are rapidly moving around, and that's because the asparagine side chain and then the glycanax is very mobile and can just freely rotate. And there are torsional angles associated with the glycans where you have freely rotatable bonds, so the sugars can move as well. Okay, so again, this might look like something like this for SARS-CoV-2 spike protein. We've already covered much of this on the slide. So, to get to the point, viruses use this. And viruses cover themselves in glycans so that you can take that static structure and then see the fuzzy shield structure. And that fuzzy shield structure allows the protein to hide from the viral or from the immune system. So shown here is the density of glycan shields and how high monos they are. You see that things like HIV and SIV, the monkey version of HIV, are very much hidden by their glycan shields, where things like coronaviruses are not that hidden from the immune system. Whereas things like flu and Lassa, if you've ever heard of Lassa, are also pretty high in this kind of immune evasion. And what that might correlate with is the sites of glycosylation, and you can see on all of these different densities, where you can see, for example, HIV. This is one we showed earlier. These are these static pictures. When they start to move around, you start to hide a lot of the surface. So glycans can play roles in biological systems, such as hiding from the immune system, but they can also play structural roles. And what do I mean by that? So this is some work out of Romy Amaro's group here at UCSD using molecular dynamics.
So would parasite, like animal parasite proteins also be like more glycosylated than proteins?
No. And that's because the glycosylation is different. And that in the lower complexity organisms, there's often not as much percent of glycosylation. Viruses take their glycans from the host. So they use our glycans to hide from ourselves. Make sense? Okay. So Romy Amaro's group looked at this glycan in SARS-CoV-2 and showed that it actually works in a structural role, where you can see it shown here in blue and green, and props up part of the protein. So this is the receptor binding domain of spike. It goes from a closed state to an open state and swings up, and then these glycans swing under to prop it up and keep it up, and thus make the virus more infectious. So this is an infection representation, and we can see a little more of this density here. But this actually happens for proteins in human biology as well, not just infection. There's also another example where this acts as a gating mechanism, that a glycan has to move out of the way for this transition to happen. So you have these structural regulation of protein biophysics by these glycans. Okay, so we've talked about glycosylated proteins, we've talked about glycotypes. Let's talk about lectins. Lectins are broadly defined as any protein that bind to sugar. So shown here are 3 different structures of lectins. These all bind high monos glycans, but they can bind them in different ways and in different types, or at different residues. So for example, glycine here recognizes these terminal 3 sugars, and you can see they kind of fit in there like a screwdriver at the top of glycine. Cyanovirin N recognizes these disaccharide arms that kind of lay through a central groove right here. My favorite lectin, and this is actually one we work in my lab, OAA, recognizes recognizes this core structure of bond 5 here that sits kind of perfectly across the top of this beta barrel. So they have different contacts with different sugars, and this is shown for high-monos glycans, but they can be extended between. There's lectins for O-link, specific O-link structures, for specific N-link structures. For any structure that exists, there's probably a specific lectin we bind to it. So carbohydrate structures are recognition sites for lectins, and each lectin recognizes specific carbohydrates. And then all known organisms have lectins of some sort. So they bind carbohydrates with high specificity, but moderate affinity. So they don't have very strong interactions with carbohydrates, but they have highly specific interactions. Does that make sense? Then they use this term called multivalency and avidity to make high affinity interactions. So what that is you notice that if the protein structure is here in gray and the carbohydrates are in green, that each protein has 2 regions of interactions with these carbohydrates. So there are 2 binding sites. And that changes your interaction stoichiometry. So that if you have one glycosylation site, let's just say we draw this with a pitchfork, right? And a protein comes in and interacts with it. And let's say that this is the binding site. And it has another binding site over here. When this interaction is done or dictated by its K off, or it's no longer binding because we're in equilibrium, it starts to dissociate away. But instead of getting fully away from the glycan, it actually rotates to the other binding site and rebinds through an interaction with the other binding site. And this is a statistical rebinding event that occurs because you have another binding site that's close to its ligand. And thus you get an enhancement in the affinity. Does that make sense? Okay. So, a classic example of this is from influenza. Influenza is one of the best studied lectins. And that's because the hemagglutinin on influenza is one of the best studied lectins. So influenza binds to cell surfaces and interacts with sialic acids. And it does that through binding sites that are at the top of this protein. It does so in a linkage-specific manner. As you can see, the sialic acid fits into this binding site perfectly. It has to be positioned in a way that if you have the wrong linkage, you're not going to have a proper interaction. So we can have something like an alpha 2-3 linkage, which is shown for this one, where, let's orient ourselves here. Here's this side chain here. Here is the oxygen. So then our anomeric carbon will be off of this residue right here. You can see if you have the wrong linkage, you might have a steric clash with the protein, versus a different linkage will present the sugar in a way that doesn't have a steric clash. So a very common sialic acid is alpha 2-3 linked sialic acid. Alpha 2-3 linked sialic acid is the most common in human biology. It lines many of our exterior membranes, especially the mucosal membranes in our GI tract and in our upper respiratory tract. There are also alpha 2-6 linked sialic acids. These are heavily present in avian species. So we're now moving which of the sugars are linked to it. And now, for example, going back to here, let's find where that the anomeric carbon we'll put for 6 would be about here, and then you can position it differently, whether it's coming out this way for 2-3 or down into this loop for 2-6. So you can see how you can have specificity to a receptor based off of this linkage. This is also the basis of whether avian influenza jumps to humans and kills us all. So avian influenza or H5N1s have not been immunogenic in humans very much. So when we hear about all this bird flu stuff, think about COVID, but 5 times worse. And that's all dictated based off of this chemistry. Okay, So as I said, these interactions are weak. If we were to measure a single sialic acid binding to this HA, you would have a millimolar interaction. Alex, how does a millimolar interaction drive infection? Okay, well, there's 3 binding sites on each influenza. So let's say that gets us down to a micromolar. Is a micromolar enough to then drive membrane fusion? Probably not. So influenza has 3 binding sites on each hemagglutinin, but if we were to look at a virion, and here's a cryo-electron tomography reconstruction of a virion, each of these little dimples in green is another hemagglutinin. So there's something like 300 to 500 HAs per virion. And then this essentially forms a Velcro-like interaction, that the sum of these weak interactions form a strong interaction, and that is the molecular basis of avidity. So if we think about it in the context of this statistical rebinding, now if we do that all along the surface, where we have a bunch of these little pitchforks, right? I'm going to draw this all out, but I've already gone far enough. And your influenza will come along, and it'll bind, kind of move along to interact with multiple ones at once, thus having this Velcro-like interaction to provide a strong interaction. Does that make sense? And this is all carbohydrate-mediated interactions. Okay. So now that we've talked about how these interactions occur, let's talk about some other cool things with these. We have on our cell surfaces a ton of these sugars. We have this network of these proteoglycans, which are essentially these disordered proteins extending up from the cell surface. And these are all heavily decorated in sugars, where you can see in this representation, these sugar chains are coming off the sides here, but you have the proteoglycans coming off the cell surface. And these form a structure that we refer to as the glycocalyx. And the glycocalyx is truly, in my opinion, the last frontier of the cell, where we actually kind of have this fuzzy layer formed by these structures that are then extending from the cell membrane. I felt kind of lied to by the textbooks when I learned about the glycocalyx. Because for a while I thought that, oh, on a cell membrane, you just have a bunch of like membrane proteins hanging out, right? It's some lipids. No, but it's actually this kind of dense forest of these glycopolymers, and that proteins have to exist and do biophysics and biochemistry in these environments. Importantly, these also act as a barrier, because in order for influenza to get to the cell and to infect the cell, it has to get through this glycocalyx. Unfortunately, viruses have evolved to do this quite efficiently, so that's probably not a great example. But there are a number of other pathogens that can't get past certain cell types, glycocalyces, and thus get stuck and do not have productive infections. Okay, so the components of the glycocalyx are predominantly GAG polymers. GAGs are glucosamine glycans. And there are these 6 different disaccharide linkages that then repeat in these very long structures. So, has anyone heard of hyaluronic acid before? I believe it's like face cream stuff. Or heparin sulfate, the anticoagulant. What about chondroitin? These are all things that are often used in the pharmaceutical industry or in the cosmetic industry for a variety of applications, so you might have heard of them, but it's because they make up the cellular glycocalyx and support those structures. Importantly, you have some sort of glucosamine attached to the reducing end sugar, and then they have some sort of charge associated with them, where most of them are sulfated in different patterns, except for hyaluronic acid that just hangs out here with carboxylic acid. Okay. They get attached to proteins in these chains, and those are GAG chains, and they can be represented by things like heparin sulfates or kerosene sulfates, shown here. So, another rather famous scientist at UCSD proposed some really interesting bases for GAGs and what they do. This gentleman was Roger Shen, who said that very long-term memories can be stored in patterns of holes in perineuronal nets. Well, it turns out that perineuronal nets are just chondroitin sulfate proteoglycans. And that what they do is if you see a nerve cell, they actually make structures along these, and they're labeled here in green, or sorry, in red. You can see these different red patches. They are these perineuronal nets that support neurons and neural synapses. And what was found later on is that the sulfation pattern between something like a 4-OS sulfated chondroitin sulfate in this versus a 6-OS sulfated chondroitin will change how mice behave and how they make memories. And that you can actually, have a subject mouse and a stimulus mouse and separate them from each other and do these hardcore neurophysiology readings that I don't understand. But see that the control mice that have the proper sulfation pattern work one way, but then there's no change when you have the wrong sulfation. So the behavior that you're training into the mice doesn't occur. There's no memory formation if you don't have the right glycan pattern. Okay, so in the last few slides here, I've talked a lot about proteins being glycosylated, because again, I'm a protein chemist that works on glycans, but other macromolecules can be glycosylated. And I say this field is in its infancy, because only, what is now 2 years ago, did we find out that RNA molecules on the cell surface get glycosylated. This is some work from Ryan Flynn's lab at Harvard that found glycoRNAs, where these specific glycan structures get added to a sugar hydroxyl group in an RNA structure, which would probably be